Hello, welcome everyone. This is chapter 3. We're looking at organic molecules this week, and we're just going to get started, okay? So we start with carbon. Carbon is central to life. Um, Anytime that we talk about life, we say that it's carbon-based chemistry, okay? Uh, uh, Questions that are never asked is why? Why is life carbon-based? Well, it's central because we need an atom. We need an atom that will allow us to make as many diverse molecules as we possibly can. Does that make sense? Life is very complex. If life is going to be complex, then we need an element that will allow us to do complex things. So if you remember to last week when we were dealing with the periodic table, okay, and we talked about valence electrons and how hydrogen has one valence electron and helium has two, and they're part of the the nobles here that don't want to play with anybody else. Lithium has one, beryllium has two. So lithium, it plays with one atom. Beryllium plays with two. Boron has three valence electrons, so it plays with three. Carbon plays with four other atoms. It can make four valence covalent bonds, and that is the maximum number. As we move on to nitrogen, we decrease by one. When we move over to oxygen, we decrease by another one. When we go down to fluorine, we decrease by another one. And when we get to neon, there's no chemistry going on anymore. So the optimal element, if you want a highly variable element that can make the maximum number of bonds in order to deal with this complex subject that we call life, we have to deal with carbon. Okay, so that's a number one answer for everything. Okay, um, so the complexity of living things is facilitated by carbon's linkage capacity. Carbon has a great bonding capacity due to its structure. Okay, carbon's outer shell has only 4 of the 8 electrons necessary for maximum stability in the elements. Okay, so again, it can make up to 4 bonds, and it makes very stable bonds. At room temperature. Uh, why does that matter? Well, it matters a lot. Let's say, for the sake of argument, you're paying attention to this and you go, well, Eleazar, if what you're saying is true and the maximum number of bonds is the only thing that matters, then why do we use carbon? Why don't we use silicon? And the truth is that it's true. Silicon can make 4 bonds just like carbon. But where do we find silicon? Look out your window. Okay, your window is silicon dioxide. At room temperature, silicon does not carry out good chemistry. It is stuck. It is. Can we have chemistry with silicon? Yeah, if we raise the ambient temperature a few thousand degrees, right? But carbon carries out these reactions at room temperature. So not only is it the most variable element, not only does it form stable covalent bonds, but it does it around the temperature that you and I evolved, which is room temperature, 20 degrees Celsius, 72 degrees Fahrenheit. Okay, so these are important things. So stable covalent bonds with a wide variety of atoms, including other carbon atoms. As a matter of fact, this variability is so high that carbon can make what's called isomers. So if you look up here, this is butane and this is isobutane. They're two completely different molecules, okay? but because of the number of bonds that carbon can make, which is 1, 2, 3, 4, we can actually make two diverse molecules with different chemical traits from the same number of carbons and hydrogens. When this happens in life, they're called isomers. They have the same chemical formula, but they have a different arrangement, okay? So that's carbon. Now we introduce ourselves to functional groups, okay? Groups of atoms known as functional groups can confer special properties on carbon-based molecules. So functional groups, I'm going to be sincere, what it adds is some degree of polarity to carbon, okay? Most interactions between molecules, most interactions in chemical reactions are driven by polarity. In other words, how negative one molecule is versus how positive another molecule is. And so the degree of polarity matters. So when you add a group, a set of atoms that confers a certain polarity, that is called a functional group, okay? So groups of atoms known as functional groups can confer special properties. What kind of special property? Polarity. They can also confer the absence of polarity, okay, or hydrogen bonding. All these things are possible. Special properties on carbon-based molecules. For example, the addition of a hydroxide group, OH, to a hydrocarbon molecule always results in the formation of a special type of molecule called an alcohol, okay? As it turns out, it also forms another type of molecule called a sugar. So sometimes alcohols and sugars are so similar that even scientists can't agree on which one they are, okay? So what happens? This just made the hydrocarbon polar. It gave it a polar character. Hopefully you remember from your periodic table that oxygen is a very polar element because it's towards the upper right, and consequently it pulls electron density away from the hydrogen, and this has a partial negative charge on it, and that instills certain traits on the carbon that now has this hydroxyl group, okay? So here's other functional groups. This is called a carboxylic acid. So it's a hydroxyl group and a double bonded oxygen. This whole thing is called a carboxyl group. Hopefully you can guess that if a hydroxyl group is polar, that a carboxylic acid is even more polar, okay? Where do we find these? We find these in fatty acids, and we find them in amino acids. Notice that every single thing on this chart is in red. That's because the position of these functional groups and where they are found is very, very important in this chapter, okay? Here's a hydroxyl group. A hydroxyl group is an oxygen and a hydrogen, okay? Look at this. Where are they found? They're found in alcohols and they're found in sugars, okay? So if you see a hydroxyl group, you might have ethanol, which is an alcohol, or you might have glucose, which is a carbohydrate. An amino group is a group that has a nitrogen to it, okay? This is a very important functional group, and it's part of amino acids. Amino acids are precursors to proteins, and proteins are incredibly important in life. Finally, we have what's called a phosphate group. Look how negative this guy must be. I mean, here's a hydroxyl group with one oxygen. Here's a carboxyl group with two oxygens. Here's a phosphate with 4 oxygens. Do you think this guy is negative? It is incredibly negative. So when you find it in molecules such as adenosine triphosphate, where we have not just one of these guys, but 3 of these guys linked like this, they are not happy. They're not happy because you have a negative charge right next to a negative charge, and they do not want to be next to each other. What are we describing? We're describing potential energy. Imagine you have a spring and you put a whole bunch of pressure on that spring, and you cock it together. What do you have in that spring? You have potential energy just waiting to be released into kinetic energy. That's what this phosphate does in adenosine triphosphate. These three phosphates store incredible amounts of potential energy when ATP is hydrolyzed into AMP, adenosine monophosphate, and these phosphates are released. That potential energy is converted to kinetic energy. Very, very cool. Also, it's a precursor to DNA and RNA, deoxyribonucleic acid and ribonucleic acid, the genetic code. Why? Because this guy builds what's called the phosphodiester backbone of these two polymers. So if it were not for this phosphate group, one nucleotide would not be able to link to the next nucleotide. So very important. So it's important for you to remember what they're called and where they're found, okay? Okay. So now we're gonna look at each one independently, okay? We're gonna start with the easiest ones, carbohydrates. Not to be confused with hydrocarbons. Carbohydrates are sugars. Hydrocarbons are oils, okay? So carbohydrates, okay, the monomers are called simple sugars. So those simple sugars can be glucose, they can be fructose, okay? Simple means that it's a monomer or at its most complex, a dimer, two sugars. Those are simple sugars. These monomers can be linked to form larger carbohydrates or polymers, which are known as polysaccharides. Poly means many, saccharide means sugars. So a polysaccharide is a linkage of many sugars. These polysaccharides are also known as complex carbohydrates. So again, when you have a single sugar or just a couple sugars, those are simple sugars. If you tether them all together into a long chain, we call those complex carbohydrates. Now, it's important for you to keep in mind which one of these organic molecules are polymers and which are not polymers. Carbohydrates are polymers because there's many, many sugars linked together to form a polysaccharide, okay? Frequently, carbohydrates have the following ratio of elements: carbon, hydrogen, and oxygen in a ratio of a 1 to 2 to 1. What is this telling you? This is telling you that we're going to have 1 carbon to 2 hydrogens to 1 oxygen. What is this trying to tell you? This tells you that this is trying to tell you is that sugars really is a carbon with a water molecule attached to them. That's what it's trying to tell you. So all sugars have this motif of having 1 carbon, 2 hydrogens, and 1 oxygen. 1 carbon with a water molecule, okay? I'm forgetting to say something that's important about this. What are carbohydrates used for? They're used for energy, right? And they're also used structurally to keep things together, okay? What are the 4 main polysaccharides? Well, there's starch, glycogen, cellulose, and chitin, okay? If you're eating a delicious little potato, you're eating starch. The way that plants store carbohydrates is in the form of starch. Animals, cows, animals such as us store complex carbohydrates as a glycogen. These two are almost identical. They're almost identical. The main difference is that because we are more demanding than plants, glycogen has a tendency to be more highly branched, okay? So that when the enzymes come in and they turn this complex carbohydrate into simple carbohydrates, so that you and I can do something demanding, something energetically demanding, okay? More simple sugars will be yielded from glycogen than from starch. That's okay. As soon as we eat starch, our body turns this starch into either glycogen or fat or energy. Okay. Cellulose. These are structural. Cellulose and chitin is structural. So whenever you're looking at plant material, it's cellulose. Okay. They tell you to eat a high fiber diet. What they're telling you to do is to eat a diet that is rich in cellulose, which is funny because we can't digest cellulose. We don't have the enzyme to recognize it. It literally goes through our entire digestive system. And that's really why you're supposed to eat it. Because if you eat a diet that's rich in cellulose, that fiber cleans out your digestive system and it's healthy for you. It actually inhibits the creation of cancer in the colon, which is incredible. So, but if you want to see, if you can't picture what 100% cellulose looks like, open a bag of cotton balls. Cotton balls are almost 100% cellulose. And if you want to be scared a little bit, there's some crazy people that are trying to lose weight and they went on the cotton ball diet. I strongly do not suggest this. Okay. But the point is that this is plant material and we actually kind of eat it every day. It's just that we can't digest it. We can't break it apart. Okay. Chitin, same concept. This is structural, but we find this in insects. Okay. You want to hear something disgusting? Look on your bag of legumes, your beans, your rice, your lentils, and chitin is one of the ingredients. Do you know why? Because when they're harvesting this, insect parts are being harvested as well, and they end up as being part of your food. Yummy, huh? Anyways, chitin is another type of a complex carbohydrate, and it just has a different uh, um, addition to it that again our body cannot recognize, and so we cannot digest it. Now, having said that, you know the funny part is that cute little cows and horses can digest cellulose. Okay, so if you give hay to a cow or to a horse and they eat it, they will still stay alive because they have the enzyme to break the cellulose into simple sugars. You and I do not. If you and I eat hay, we're going to die because we don't have the enzymes to break this apart. Is that weird? So here's starch, here's glycogen. Remember I told you that glycogen and starch are almost identical. The main difference is that glycogen is more highly branched. Here's starch in a delicious little potato. Glycogen, your author wants you to know that glycogen is stored in the liver. The truth of the matter is that we all have about 1 kilogram of glycogen in our bodies. Half of it is stored in the liver, the other half is stored in the muscles. Okay. Why? Because when you need to do something, when you're in a fight or flight response and you need to run away from Wrinkles the clown or whatever, okay, the glycogen in your muscles is going to provide energy first, and then the glycogen in your liver is going to follow up on that. But your author doesn't want us to know that for some reason. So for you and me, in this book, glycogen only comes from the liver. Okay, whatever. And then here's cellulose. Okay, this is also sugar, but it's got a different linkage on it, and because of that linkage, we cannot digest it. So this is structural. And here's chitin in our delicious little insects, right? Insects. And that's also, we cannot digest that either. So both of these are structural and these are for energy. Okay. Okay, glycogen is a nutrient storage form of carbohydrates, glucose, in the liver of animals. Liver. Cellulose is a rigid structural carbohydrate found in the cell walls of many organisms. Structural. Chitin is a tough carbohydrate that forms the external skeleton of arthropods. Those are called insects. Chitin, by definition, is a structural carbohydrate. Structural. Okay, we're switching to lipids. So, the defining characteristic of all lipids is that they do not readily dissolve in water. What is that called? Do you guys remember? That's called hydrophobic, water fearing. So all lipids are hydrophobic in character. That is their main characteristic. Lipids consist of triglycerides, okay, steroids, where steroids consist of cholesterol and hormones. Lipids also consist of phospholipids and waxes. So these are the four main categories of hydrophobic organic molecules that we care about. Okay, lipids do not possess the monomer to polymer structure. Remember, you have to keep track of which have polymer character and which do not. Lipids do not. Okay, and we're going to see why in just a second here. Let's see. Among the most important lipids are the triglycerides, composed of a glyceride and 3 fatty acids. So it looks like this. Okay, this is a triglyceride. So this 3 fatty acids, 1, 2, 3, and then our glycerol. Your author calls it glyceride, glycerol, potato. Your glyceride or glycerol backbone right here. So this is a sugar, because this has a little H here. You don't see it because we got rid of it. But this is a hydroxyl, this is a hydroxyl, and this is a hydroxyl. So this is a sugar or an alcohol, glycerol, right? Remember, we have a hydroxide. It can be either a sugar or it can be an alcohol, okay? And then these are fatty acids, okay? Picture an OH group right here, okay? So this functional group is supposed to be a carboxylic acid before this thing is formed, okay? So this is a triglyceride. Why do we care? Because when you and I store fat, this is the way that we store it. When our cute little cows that we eat store fat, this is the way that they store it. So if you're eating a steak and it's not well marbled and you're consuming fat from that steak, you are consuming triglycerides. Okay, so this is a main source of energy from fat, a triglyceride. Okay, switching to another fat, the steroid. Another important variety of lipids is the steroid, all of which have a core of 4 carbon rings. Examples include cholesterol and hormones such as testosterone and estrogen. So this is very interesting, okay? See the 4-ring motif? 1, 2, 3, 4, okay? This is a steroid, but this steroid can function as a messenger, such as testosterone in men and estrogen in women, or it can function as a means of breaking up plasma membranes as cholesterol. So this has a lot of roles, okay? It can function as a messenger, or it can function to disrupt plasma membranes, okay? Switching to phospholipids. A third class of lipids is a phospholipid. These are composed of 2 fatty acid glycerols and a phosphate group. This is the material that forms the outer membrane of cells and which is largely composed of phospholipids. Notice I have 2 fatty acids here, okay? The reason I have that highlighted is because people love to confuse the fact that triglycerides have 3 fatty acids with the fact that phosphates have only 2 fatty acids. So you can't confuse that. Tri is 3, triglyceride, 3 fatty acids, 3, and then phospho, 2. Okay? Can't confuse those two. So here's a phospholipid. Okay? Here's our 2 fatty acids, one here, one here. We have a glycerol backbone, no surprise. We have a phosphate group, no surprise. And then we have what's called a variable group. This variable group is a sugar group. And this is the part at the top of the plasma membrane that lets your cell tell everybody else, hey, this is my ID card, just letting you know I belong here. So this variable group is a sugar that your body imparts to all of your cells so that those cells can tell everybody else, I belong here. Because if that variable group is not recognized, your immune system will come over and they will kill that cell. They sure will. Okay. Now, this is an experiment to demonstrate what happens to what's called a membrane leaf. So a lot of people forget that a plasma membrane is not 1, but 2 phospholipid layers. Okay. So there's a hydrophobic. Let me try that again. There's a hydrophilic, one to the outside. There's the lipid tails, and then those lipid tails go to another set of lipid tails, and then there's a hydrophilic heads to the inside of the cell. So you have 2, and each one of these is called a leaf. So this is a leaf right here. See that? And if you were to throw that into water, the polar heads would associate with the water, and the hydrophobic tails would associate with each other. So something. There's a way that the leaf works. Waxes. I don't want to spend a lot of time on waxes. The main significance about waxes is that they are in fact a lipid, and they're used usually structurally to protect an environment from water. Okay, so it's a cuticle of a plant, and you can see how thick the wax layer is, preventing water from not really entering, but preventing water from escaping the cuticle. Okay, so that's important. One last concept, one last point. Your author kind of snuck this in, which is great. Uh, Mr. Krogh wants to talk about the hydrogenation of vegetable oil. Okay, so you're trying to be a healthy college student. You're trying to exercise. You're trying to eat right. Okay, and you know that if you eat fats that are poly unsaturated, that they're better for you than fats that are saturated. Everybody knows that, right? Now, the first question is, what does saturated mean? What are we saturating with? Well, this is what we're saturating with. We're saturating with hydrogen. See what happens to here's my fatty acid. See my carboxylic group right here? Here's my fatty acid, and it's saturated, which means I cannot add any more hydrogen. This is it. What happens to the shape of this fatty acid when it's saturated by hydrogen? And the answer is that it becomes straight, just like that. Why is that bad? Because if you have a double bond in fatty acids, it disrupts the shape of the fatty acid. You see this? It places a kink in it. So in your plasma membrane, because this will eventually go through your plasma membrane. If you have a whole bunch of these guys that are completely straight, they're going to be like this, just like that. And they're not going to want to pull away from each other. It's a very weak interaction. It's called van der Waals. Okay, it's called a van der Waals interaction, and it's incredibly weak. But for a very long lipid, such as this one, weak interactions become very powerful. And what happens is that your plasma membrane ceases to be dynamic. It starts getting stuck like this all the time. Do we care? The answer is yes. Because in order for a cell to be dynamic, in order for a cell to do what it needs to do, it must take energy in, and it must get rid of waste, and it must release hormones and receive messengers. In other words, the only way that a cell communicates with its environment is through its plasma membrane. And if you have a plasma membrane that's locked up with these saturated hydrocarbons, that cell is not going to be able to communicate, get energy, get rid of waste, do those things that it needs to do in order to carry out its normal function. But if you have a plasma membrane that is made out of poly, this is not poly because it's only a single double bond. Here's a poly. Poly means many, right? Polyunsaturated, which means that it has many double bonds, then a whole bunch of kinks are going to be created. And now, all of a sudden, your plasma membrane, these things can move around a little bit. They can move around. They can move around. And because they can move around, things can come in easily and things can leave easily. And your cell can communicate without difficulty, etc. Does that make sense? Now, here's the problem. When you have people in business that don't have scruples, they're pretty much out to kill all of us, okay? So, you and I are looking for fats that are fully unsaturated because we know that that's supposed to be good for us. And what do these guys do? They pull it's a switcheroo, and they say, hey, here's this thing that's really good for you because it has polyunsaturated fats. Oh, incidentally, it's hydrogenated, made out of hydrogenated vegetable oil. What are they telling you? What they're telling you is that they started with something that was polyunsaturated, and then they hydrogenated it. In other words, they added hydrogen to turn it into this stuff. So if you have hydrogenated vegetable oil, the only thing that you read is polyunsaturated fats, and you think you're buying the right stuff. No, you're buying terrible stuff. You're buying what you're not supposed to be buying. So this is false advertisement, okay? So the hydrogenation of vegetable oil yields a decrease in the number of double bonds, turning something good, polyunsaturated fats, into something bad, saturated fats. Nice, huh? Okay, switching to proteins. Um, proteins and nucleic acids. That's really where it's at. If you want to know how something lives, you need to learn about proteins and you need to learn about nucleic acids. Okay, first of all, the monomers contain both an amino group and a carboxyl group. Okay, fortunately, there's one up here. Here we go. So here's an amino acid. Here's the second amino acid. Here's the third amino acid. And we can see is that each amino acid has a central carbon, has a hydrogen, has an amino group, and has a carboxyl group. Do you see that? We go to the next amino acid. We see that it has a central carbon, a hydrogen, an amino group, and a carboxyl group. Here's the third amino acid, central carbon, side hydrogen, amino group, and a carboxyl. So this is an amino acid. The only thing that changes is this side chain over here. And we have 20 different amino acids. Okay, very, very cool. So amino acids contain both an amino group and a carboxyl group. Sequences of amino acids are strung together to produce polypeptide chains. So again, this is a polymer. Poly means many, peptide means protein. So polypeptide chains, which then fold up into working proteins. Important groups of proteins include enzymes, very important, which hasten chemical reactions. Let's face it, guys. At the end of the day, we're trillions upon trillions of chemical reactions. And furthermore, we're demanding animals. We want everything done now. So how can we have that? We have to have enzymes that facilitate or speed up these chemical reactions. Consequently, we have a lot of enzymes in our body. Okay. Structural proteins, which make up structures such as hair, fingernails, your keratinized skin, bones, ligaments, tendons, the keratin of your eye. All these are structural proteins, proteins that hold your body together. Okay. So look at this. See, up until now, everything did only a couple things, right? So for example, if you're dealing with sugar, that's energy or structural, right? If you're dealing with lipids, it might be communication, energy, and plasma membrane, right? Not a lot of things. You get to proteins, proteins do everything, okay? So they can be enzymes, which speed up chemical reactions. They can be hormones, which means that they function as chemical messengers, okay? They can carry out transport. So here your author gives you an example of hemoglobin to transport oxygen. But honestly, walking on your legs is also a type of transport that's carried out by proteins. Contractile movement, myosin and actin, which allow muscles to move. Okay. Protective, so healing defenses against an invader. I like the example of antibodies. Okay. So our bodies are protected by antibodies, which are again proteins, protective. Structural, okay? So keratin, hair, collagen, cartilage, all these things that hold our body together. Structural. Storage. On top of all this is storage. What does that mean? That means that once we run out of sugar, we switch over to oils, to fats. And when we run out of fats, we switch over to protein. So if you don't have enough sugar or enough fat to keep you alive, protein can be another source of nutrition. Um, Toxins. So when snakes make poisons, for example, those are proteins. But really, your white blood cells produce toxins to help your body defend against bacteria. Okay, so that's incredible. Communication. It allows cells to communicate with each other, and allows your cells to communicate together so nicely that you can think, and you can walk, and you can draw conclusions, and you can get bored in a biology lecture. How is that cool, right? Okay, so enzymes, it allows you to do a lot of things. Okay, again, proteins are the most complicated of all because there's a hierarchy to the structure. There's what's called the primary structure of the protein, which is the amino acid sequence, okay? So, for example, here we have the amino acids alanine, leucine, serine, glutamine, histamine, alanine, on and on and on. This is the amino acid sequence, okay? And that is called the primary structure of a protein, okay? This primary structure, this sequence of amino acids, determines or dictates the secondary structure, okay? What does secondary structure look like? It looks like this. So here's primary, here's your amino acid sequence, here's your secondary structure. Motifs such as alpha helices, so here's an alpha helix, okay? Uh, pleated beta sheets, random coils, okay? And don't think because they're called random coils that they're not important. In your immune system, antibodies use this random coil to identify different molecules. So this random coil, it can be very important, okay? So to recap, the amino acid sequence, which is called the primary structure, dictates whether this thing bends into an alpha helix or a beta plated sheet or a random coil, which is called the secondary structure. These will then fold onto each other, okay? These motifs may persist through a set of larger scale turns that make up the tertiary structure, third degree structure, okay? Then finally, when two or more of these guys come together, they come up with what's called the quaternary structure. So I recognize it. The author's doing hemoglobin here. So we have two alpha and two beta subunits, which makes hemoglobin, okay? And this is a quaternary structure of hemoglobin. So you just need to remember, primary structure is amino acid sequence. Secondary structure is pleated sheets, alpha helices, random coils. Tertiary structure is the folding of these into a large subunit, and quaternary structure is the association of two or more subunits to form a quaternary holoprotein. Very, very cool. Lipoproteins. This is one of my favorite topics. Okay. You guys are probably too young to care. I'm more than old enough to care. But we deal with these things called high density and low density lipoproteins. Okay. When you go to your doctor, these are called the good and the bad cholesterol, respectively. Okay. So the good is high density, the bad is low density. And it says which transports cholesterol in human beings and are important determinants of human heart disease. The truth of the matter is, ladies and gentlemen, is that the liver dictates the production of these, okay? And if you're a sedentary person, which means that your definition of exercise is looking for the remote control when you lose it, and all you do is eat potato chips, then when triglycerides, cholesterols, or other lipids come into your intestine, your liver goes, all right, guys, come on in here. We don't really have need for you, but we're not using you. So go ahead and come on in. Okay. However, if you decide to walk, stroll, take a constitutional, jog, play basketball, any of these things that cost exercise, all of a sudden the body goes, wait, what's going on here? Apparently, we're using energy. So now we have to go from our fat stores and transport this from the fat stores to the liver to that part of the body that's using them, okay? So in other words, LDLs take fats from the outside and assimilate them into the body, and HDLs take fats from the body and eliminates them, either because they're being used as energy or because they're being eliminated into the intestine. So, not surprisingly, the more exercise that you do, the more HDLs that your liver produces. And the more HDLs that your body produces, the more that nasty types of fats will be taken from your body and thrown out. Why? Because we're not going to build all these HDLs for nothing. Once we produce them, they need to be doing a job. And if that job isn't transporting fat to where they're going to be, to where it's going to be used, we're going to transport fat to where it can be lost. And consequently, people that exercise regularly, and I'm not talking vigorously, I'm just talking moderate exercise, can have a high HDL, which is considered protective. Protective of what? The answer is protective of your heart, because saturated fats are being taken away from uh, um, arteries in particular, and some veins, and is protecting the health of your heart. And the thing that we have learned recently is that if you are a young man and you have thrown, or woman, I say man because usually guys are the ones that really don't care. If you've been a young man and you've thrown care to the wind and you've eaten cheeseburgers your entire life, and now you find yourself in poor health because you've been doing nothing but consuming a diet high in saturated fats, at any time you can stop, change your diet into a low fat diet, start exercising, and your body will literally reverse all these problems. We thought that these cardiovascular problems were non-reversible. That is not the case at all. There's no point at which the body, well, there is a point. We call it death, right? Once you're dead, kind of hard to exercise then. But while you're alive, there's no point at which your body cannot take advantage of this and go back to pristine health, which is beautiful. Okay. Okay. What will pH do to a protein? Will pH do something to a protein? Yeah, it will. We have various environments in our body that only function at either low or high pH. So a good example of a low pH is our stomach and our duodenum, because it is where the chyme that still has hydrochloric acid enters the small intestine. Okay, so consequently, what evolution has done is that the enzymes that enter environments of low pH, they enter in a non-functional form, and the pH causes something to happen to the enzyme in order for it to become activated, which is very cool. And this is not only true of digestive, well, it's mostly true of digestive enzymes. So a change in pH can stop the function of an enzyme if it's not supposed to be functioning in that environment, or it can promote the function of an enzyme. It can change the shape of the enzyme. So in fact, that's usually the way it does is by cleaving, okay? It can change the binding properties of the enzyme for the substrate. In other words, that substance upon which the enzyme functions. It can change the tertiary structure. You remember what that is, right? Alpha helices and random coils and beta plated sheets. It can change the quaternary structure. This is usually where it happens. Subunits are literally cleaved from each other. What it does not do is it does not change the amino acid sequence, okay? Unless you have an enzyme like protease, whose sole purpose is to take a protein and break it apart into its individual monomers, which is amino acids, a change in pH will not change an amino acid sequence, okay? But it can do all these other things listed here. Okay, this is the last part, nucleic acids. They're polymers. Polymers of what? They're polymers of nucleotides. So the monomer is a nucleotide, and the polymer is called either DNA or RNA. Okay. And here we're going to talk about the probability of nucleic acid. Oh, I don't know, because it might come up somewhere. Now, we're going to go into this in more detail in future chapters. However, nucleic acids make up words of three letters. Okay. So the reason that we're discussing this is because once upon a time, scientists believed that the genetic code was made out of amino acids and not nucleic acids. And the reason for that is because there's 20 amino acids and only 4 nucleic acids. So it's hard for scientists to believe that something as complex as life could depend on only 4 letters. It just didn't seem right. So when you can do the math, we now know that genetic material is dictated by a letter called a codon, which consists of 3 letters. So my question to you is that if you have a codon consisting of 3 letters and you have 4 nucleotides, how many different words can you make? Follow me on this one. In the first position, we're going to have 4 different nucleotides. We're going to multiply by however many nucleotides we can have in the second position, which not surprisingly is 4. 4. That didn't do what I wanted it to do. 4 times 4. There we go. And then in the third position, where we're going to multiply by however many nucleotides we have there, which is 4. And we get 64. Not surprisingly, now we know, and you're going to learn soon if you don't already know, that this is the number of codons that we have. We have 64. Now, what does the math look like if you use amino acids? Well, how many amino acids are there? Well, there's 20 in the first position, and we'd have to multiply that by 20 in the second position, and then we'd have to multiply that by 20 in the third position. So with 20 amino acids, we can make 8,000 words. This does not go contrary to what these scientists were thinking. But all that being said, we only have 64 codons. So the correct answer ended up being 4 nucleotides in the first position times 4 nucleotides in the second position times 4 nucleotides in the third position for a total of 64 words, 3 letters long. Okay? So this is important for you to know. Let's see. It's important for you to know that DNA, deoxyribonucleic acid, is composed of nucleotides, and that each nucleotide contains a sugar, a phosphate, and 4 nitrogen containing bases. So let's see if we can look. Let's make it big, okay? So let's make it really big. There we go. Each nucleotide consists of a sugar, okay? So that's going to be, this is DNA, so it's going to be deoxyribose, phosphate group, and 4 nitrogen nucleotides, which here are going to be guanine, cytosine, thymine, and adenine, okay? What else? Notice that the molecules of DNA are anti-parallel. What does that mean? Well, they're parallel to each other, but anti, because we see these sugars are facing up, and these sugars are facing down. Do you see that? Up, down. That means that they're anti each other. And so the DNA double helix is said to be anti-parallel. And the anti-parallel strands associate with their complementary strand through this little guy right here. Can anybody guess what these are? I wish I could hear you, but these are hydrogen bonding. So it is complementary hydrogen bonding that allows the complementary strand to associate with the daughter strand. And the complementary strand, bonding, hydrogen bonding, is always going to be G to C and T to A. We keep on going G to C, T to A, G to C, T to A, T to A, on and on and on. This is called, you don't need to know it, but it's called Chargaff's rule. Okay? So they're held to the complementary anti-parallel strand by hydrogen bonding. Okay, let's see. DNA is a repository of genetic information. The sequence of its bases encodes the information for the production of huge array of proteins produced by living things. And you need to learn that these proteins are created by what's called the central dogma, which is that the DNA in the nucleus produces messenger RNA that goes out into the cytosol, and then this attaches to ribosomes to link amino acids together and make protein. So it goes DNA, RNA, protein. That's the central dogma. And let's see. We have a second nucleic acid, which is RNA, the messenger RNA. Again, that's the guy responsible for transferring information from the nucleus and from DNA. Okay. And then the last thing that we use these nucleotides for is the production of energy. So hopefully you remember that energy in the form of adenosine triphosphate. This is the currency that your body recognizes. This is the potential energy that your body uses. So this is made out of nucleotides. This is made out of nucleotides, and this is literally a nucleotide. Okay. That is it, ladies and gentlemen. I hope I didn't bore you too much. Have a wonderful day, and until I see you next. Bye-bye.